Hello, everyone. Welcome to my video tutorial for RNA differential splicing analysis. The data that I'm using for today's demonstration is the FOXP1 deficient mice. If you want to use this dataset for practice, you can download the data using the link for GSE 118617. You can see in this dataset, they extracted the basal and the luminal epithelial cells from the mammary glands of FOXP1 control mice and FOXP1 conditional knockout mice. Because we are going to do differential splicing analysis, so we need to start from the original sequencing data. If you click the link edgeR run, then we can find all the FASTQ files for this dataset. You can see here we have 12 samples here, 6 samples for basal epithelial cells and 6 samples for luminal epithelial cells. Each cell population has 3 repeats. You see control knockout, control knockout, control knockout for basal epithelial cells, then control knockout for luminal epithelial cells. You can use the SRA toolkit to download all the data. For this dataset, because the size is not big, you can see the biggest one is 2.17 gigabyte. So you can directly download from NCBI website. You don't have to use the SRA toolkits. If you click the first one, then you can click the download link for the FASTQ files. Let's click this link. Then here, if you click the FASTQ file, you will download the first file for the control basal cells. So I downloaded already. You can try to download all the files in your computer. Then next we can perform sequence alignment. So I use the R subret to do the sequence alignment. Because this dataset is from mouse, so for the alignment, we need to build an index for the mouse reference genome. So we can use the mouse PRCM39 reference sequence to build the index. You can download the reference sequence from this website. Then we can use this code to build the index for the mouse reference genome. So it takes 40 minutes to build the index. I built the index before, so I can reuse it. Next, we can create target files. You can see I loaded the target files already. We can click and have a look. You see here in the first column, we have the samples information. Second column is the GSM data. But for our subret alignment, the most important columns is the input files column. You can see I downloaded the files from 28 to 39. They are compressed FASTQ files. Then you need to name output files. We named with basal control, basal knockout for repeat 1, repeat 2, repeat 3. Then luminal control, luminal knockout for repeat 1, repeat 2, and repeat 3. Once you created the target file, then you can start to do the alignment. We can use the index we built as MM39 reference. Then the read file 1 will be the input files in the targets. Because this dataset performed single and read, so we don't have the second read files. For the output files, we can use the files name in the target. Then our subret will create the BAM files for each dataset following the completion of the alignment. So this step takes about 2 hours to run. I did the alignment before, so I'm not going to show you here today because it takes too long to run the sequencing alignment. Once you have all the BAM files, you can use the feature count function to get the alignment result. So you can see here the files will be the output files that are BAM files. We use the single end alignment so the its paired end argument is false. And we use the inbuilt annotation file mm39 to create the feature count. Because we want to identify differentially expressed exons, so here for the allow multi-overlap argument, you need to set it as true, then we can get the total number of exons from all the dataset. So I did the feature count from all the BAM files and named it as FoxP1. We can connect the FoxP1 dataset and have a look at the data list in this dataset. You can see we have 4 data lists. The counts for 12 samples. In total, we have 286,508 exons. And the second list is the annotation information. You can see the gene ID, chromosome information, the start and the end position for the exons, and also the strand information and the exon length. The target list is the BAM file name. We have 12 BAM files. So now we can create a DGE list from the feature count data for the edgeR analysis. The count will be the feature count from FoxP1, and the gene argument will be the annotation information for all the exons. So let's create the DGE list for edgeR analysis. You can see we have a large DGE list here. We can use the dim function to look at the exons numbers and the sample numbers. You can see now we have 286,508 exons from 12 samples. In the DG list, we have 3 elements. The first one is the count information. The second one is the sample information. The third one is the gene information. We can have a look at the gene information from the DG list. Let's run. You can see the first column is the gene ID information. The gene ID information was created by R subarray align function. Because R subarray align function uses the gene ID information. The second column is the chromosome information. The third and the fourth column is the start and end position for the exons. And here is the strand information. You can see we have 2 genes here, 600 and 609. You can see from the first row to the fifth row are the exons from the same gene, and the last column is the exon length. Because we only have gene ID information here, we don't have the gene names. So we can get the gene names from the genome-wide annotation database for mouse. So let's load the package first. Then we can get the gene names using the map IDs function. So the database will be the mouse database we just loaded. Then the case argument will be the row names for our DGE list dataset. And we know the row names are the gene IDs in the DGE data list. Then we need to set the key type as entrance ID. And we want to get the gene names from the gene symbol. So the column argument we need to set as symbol. Then we can get the gene names for all the gene IDs in our dataset. Let's run. So before we add the gene names into the DG data list, we can have a look at the gene information in the DG data list. You can see here at the moment, in the genes data slot, we only have the gene ID information. If we add the gene names into the genes data slot for the DG list, and we can have a look at the gene data slot now. You can see we added a new column for the gene names. You can see from row 1 to row 5 are the 5 exons information for the CGLP1 gene. So we added the gene names into the DG list. Next, we can have a look at the sample information. Let's get the samples from the DG list. And we can click the samples dataset and have a look. You can see the row names are the samples name. Then we have the group column at the moment. All the groups were labeled as 1. Then second column is the library size. And the last column is the normalization factors. Because we haven't performed the normalization, so all the normalization factors is 1 for each dataset. So let's add the group information for the dataset. We know the first sample is basal cells population control. The second one is the basal cell population knockout. Then second repeat, third repeat for the basal cells. And the sample 7 is the control luminal cell population. Sample 8 is knockout luminal cells. Then the second and the third repeat. So we can set the group information. Then we add the group information in the DG list. Now we can get the samples data slot again. And we can have a look at the samples. You can see now we add the group information into the DG data list. So we have the group information. Now we can perform data filtering and normalization. We can filter the data using the filter by expression function, and we add the group information here. Let's filter the data. We can use the table function to look how many exons we should keep. You can see there are 175,404 exons were labeled as false, and 111,104 exons were labeled as true. So that's the exons we need to keep for the rest analysis. Before we filter the data, you can see in the DG list, we have more than 286,000 exons. Now we can perform the filtration, and we can have a look at the data now. You can see we only kept the 111,104 exons for our analysis. So after the data filtration, we can normalize the library size. Let's run. After normalization, we can get the samples information again from the DG list. Then we can have a look at the samples. You can see now that normalization factors were changed for each dataset. So normally we can look at the data after the filtering and the normalization. We can make a multi-dimensional scaling plot. Let's run for all the samples. And we can zoom in. You can see all the data set from the basal cell population were clustered on the left-hand side, and all the data sets from the luminal cell population were clustered on the right-hand side. From the color, you can see also the separation for the knockout samples from the control cells. For the luminal cell population and also the separation for the knockout samples from the control samples in the basal cell population. So from the MDS plot, we can clearly see there are differences on the exons expression level between control and the knockout cell populations. So we can continue our analysis. So now we can design the matrix and use the matrix to perform dispersion estimation and also differential analysis. Because we want to compare the differentially expressed exons between different cell groups, so we can design a matrix to include the group information. Here you can see we use the 0+. If we use the 0+ in the matrix design, the design will include the all the groups and doesn't have the interceptor column. Let's design the matrix and we can have a look. If we click the design, you can see here we have 4 groups: the basal cell population control knockout, luminal cell population control, and the knockout. You can see here we included the first cell group and we don't have a interception column because we want to include the first group information. But at the moment you can see the column names, we have the group in the column name. We can remove the group from the column names. We can use the G-sub function. We want to replace the group with nothing, and we want to modify the column names in the design. So let's run. You can see now we remove the group. We only have the basal control, basal knockout, luminal control, and the luminal knockout as the column names. So we designed the matrix. Now we can estimate the dispersion. And here we included the design information. Let's run. So it is done. Let's plot the biological coefficient of variance to see the dispersion for each exons. Let's make the plot. And we can zoom in. You can see the black dot show the categorized dispersion for each exons, and the straight red line show the comb dispersion. Then the trended dispersion is in blue. So now we can perform the analysis to identify differentially expressed exons between knockout and the control cells. So first we need to fit the gene-wise negative binomial generalized linear model for the Corsi likelihood test. So let's fit the model. And again, we can make a plot for the dispersion. For the fitted model, let's run. Let's zoom in again. You can see now the raw and squeezed dispersion are in black and red dot, and the trended dispersion is in blue line. So we fitted the model, we can perform the F-test to identify the differentially expressed exons. Because we have 2 cell population here, we have the basal and the luminal cell population. In the following demonstration, we are going to focus on the luminal cell population. If you want to analyze the basal cell population, you can follow the same code, just change the cell population to basal cells. So we need to make a contrast first and tell R we just want to focus on the luminal cell population. So we can make the contrast as luminal knockout cells against the luminal control cells, and the levels will be our designed matrix. So let's make the contrast. Now we can use the contrast to perform the causing likelihood F-test to identify differentially expressed exons between knockout and control cells in the luminal cell population. We can perform the F-test now. After F-test, we can use the top text function to look at the differentially expressed exons. Let's run. You can see here we show 10 exons from the analysis. You can see the coefficient is knockout cells against the control cells. We have the gene ID information, chromosome information, started and the position for the exons, strength and the exon length. We add the gene names in the gene symbol column, and here is the log FC changes. And down here you can see the log CPM, that's the count per million, the F value because we performed the F test, and the P value for each exons. And the last column is the value for the false discovery rate. So we have all the information for each exons. Now we can use the decide test function and make a summary to see how many exons are upregulated and how many exons were downregulated. Let's run this code. You can see we compare the knockout and the control in the luminal cell population. There are 285 exons were downregulated in the knockout cell population, and there are 1,203 exons were upregulated in the knockout cell population. So we identified the downregulated and upregulated exons in the knockout cell population compared to the control cell population. So now we can detect the alternative splicing by testing for the differential exons usage between FOXP1 knockout and the control in the luminal cell population. To detect the alternative splicing, we need to use the function in the edgeR package, and this function is designed to detect the alternative splicing for the genes. So we can detect the alternative splicing from the fitted model, and we use the same contrast to detect the luminal cell population. Gene ID argument will be the gene ID, and also we need the start position for the exon ID argument. So now we can run this function to detect the alternative splicing. You can see we have the information from here. In total, we have 111,104 exons, and the total number of genes are 16,118 genes. Because most of genes have more than one exons, you can see here only 4,401 genes have one exon, and the average exons for each gene is 7. The maximum number for the exons in a gene is 106. So after the analysis, we can look at the top differentially used exons. Let's run. So you can see down here we have the information for the top differentially used exons. You can see the number 1 and the number 2 is the FOXP1 exons, and they were downregulated because in the gene knockout mice, both exons were deleted. Then the second gene is the TOR1, AIP1, and the next one exons was upregulated in this gene, and the third gene is the NAS2. There are 2 exons were upregulated in the NAS2 gene in the knockout mice. You can see here all the data is on individual exons level, and also we can look at the differentially expressed result at the gene level. To look at the result at the gene level, there are two methods you can use. If we have a look at the SP result, you can see during the analysis, we already performed the analysis on the gene levels. You can see the F-test for the gene levels, and also for the second method, the CMS method on gene level. So we can use the top spliced DEG function again to look at the splicing result at the gene levels. The first one is the gene level by the F-test. According to edgeR package, F-test is more powerful than the CMS method if genes have several exons that are differentially spliced. We can set the test argument as genes. This is the result from the F-test. Let's run. You can see now we only show the differential splicing result at the gene levels. We don't show the result at the individual exon levels. You can see here, the first gene is still the FOXP1. It has 20 exons. And the second gene is NAS2. It has 9 exons. And the third one is TOR1 AIP1 gene. It has 11 exons. This is the result at the gene level by F-test. We can also see the CMS method. According to the edgeR guidance, this method is more powerful if only a minority of exons for a gene are differentially spliced. So we can also look at the test result using the CMS method. We can run. You can see here is the result from the second method. But the first gene is still FOXP1 because in this dataset, 2 exons for the FOXP1 gene were deleted. Then the second gene is TOR1 AIP1. Then the third gene is NAS2. You can see the top 3 genes are the same from both methods. And the genes down here are slightly different, using different testing method. So no matter which method we use, we can always identify the alternative splicing for the FOXP1 gene, because we know 2 exons were deleted in the FOXP1 knockout cells. So from the result for the FOXP1 gene, you can see our analysis can identify correct alternative splicing. So now we can make a plot to show all the exons for the top 2 most differentially spliced genes. They are FOXP1 and TOR1 AIP1. So we can make the first plot for all the exons for FOXP1 gene. Let's run. Let's zoom in. You can see we know there are 20 exons in the FOXP1 gene, and the exons 14 and 15 were deleted in the knockout mice. So you can see the downregulation for exons 14 and 15 in the FOXP1 knockout mice. Both exons show significantly downregulation, and they are labeled in red color. The next one we can plot the second gene, TOR1 AIP1. Let's make the plot. And we can zoom in again. You can see we know there are 11 exons in this gene, and the 5th exon is upregulated in this gene. And the upregulation is quite high because the log FC value is bigger than 3. Okay, that's it for today's demonstration. In summary, in this video tutorial, I showed you how to perform RNA alternative splicing analysis. I didn't show you how to build the index for mouse genome reference. And also I didn't show you how to align the FastQ files using R subret package. But I have video tutorials for the R subret package. You can watch my R subret videos to build the index and also align the sequencing data. So please subscribe my channel if you haven't to do so, and share my videos with your friends. Thank you.